The synthesis of the enzyme begins after a messenger RNA binds to a free ribosome, one that is not attached to a cytoplasmic membrane. As the polypeptide emerges from the ribosome, the hydrophobic ER signal sequence is recognized by a signal recognition particle. The SRP binds to both the ER signal sequence and the ribosome, temporarily arresting further translation. Binding to the endoplasmic reticulum occurs through two interactions, one between the SRP and the SRP receptor, and the other between the ribosome and the translocation channel. Once the SRP ribosome nascent polypeptide complex binds to the ER membrane, the SRP is released from its ER receptor. The ribosome becomes attached to the translocation channel, and the signal sequence is inserted into the translocation channel, displacing the plug. The polypeptide is then translocated through the translocation channel and into the lumen of the ER. Upon termination of translation and the passage of the completed polypeptide through the translocation channel, the membrane-bound ribosome is released from the ER membrane and the plug is bound into the translocation channel. As the polypeptide enters through the endoplasmic reticulum, the portion containing the signal sequence is removed by signal peptidase, and glycosylation of the synthesized enzyme is initiated by oligosacchαρyltransferase. After this, a chaperone, in this case calnexin, binds to the terminal glucose of the polypeptide in order to help it properly fold. Good. It seems it's properly folded. The chaperone then disengages from the newly folded enzyme, allowing glucosidase to come in and remove the terminal glucose molecule from the oligosaccharide. After this, glucosyltransferase comes over and checks out the enzyme to make sure proper folding has occurred. Good. It seems that proper folding has occurred. Another glucose monomer is then removed, and the enzyme is sent to the Golgi. Let's take a zoom in. Glucosyltransferase comes in after arriving at the Golgi to remove two more glucose monomers. After this, another glycosyltransferase comes in and attaches another sugar to the end of the oligosaccharide on our newly formed enzyme. Our newly formed enzyme is now ready for transport. It then enters a vesicle, and the vesicle follows along the cytoskeleton of the cell to its proper place in the cell membrane. After arriving at its designated position, a tethering protein will attach to the RAB on the vesicle. This is called tethering. Afterwards, both the V snare, the vesicle snare, and the T snare attach to each other. This is called docking. Afterwards, the T and V snares twist together closer and closer and tighter until fusion occurs between the vesicle and the cell membrane. Once fusion has occurred, this allows our newly formed enzyme to travel through the cell membrane.